Hello, everyone, and welcome to today's webinar, where we will be discussing cutting-edge LC-MS testing solutions supporting the integrity of dietary supplements. My name is Jennifer Caveney, and I will be facilitating today's presentation. Before we begin, just a reminder that this webinar is being recorded, and the recording will be sent out following the discussion. If you do have questions, we do encourage you to enter them into the questions box, and we'll do our best to answer following today's presentation. So let's go ahead and get started. I have the pleasure of introducing our speakers today. We have two excellent speakers from the Eurofins team. Our first speaker today is Grace Bandong. Grace is the Laboratory Director for Contaminants at Eurofins Food Chemistry Testing in Madison, Wisconsin. She brings over 30 years of expertise in the food and supplement industry. She specializes in elemental and pesticide residue analysis, as well as chemical risk assessment. Grace has led the development of contaminant testing programs that support supplier verification for leading food and dietary supplement brands. Our second speaker today is Dr. Lukasz Baklovic. He is the Technical Manager at Eurofins Food Chemistry Testing in Madison, Wisconsin, where he oversees contaminant and residue testing to ensure food safety through advanced chemical analysis. Previous to this role, he contributed to regulatory science at the FDA Center for Food Safety and Applied Nutrition, with expertise in chromatographic and mass spectrometric techniques. He has authored over 40 peer-reviewed publications focused on chemical residues and adulterants in food. Dr. Vaklovic holds a PhD in food chemistry and analysis from the University of Chemistry and Technology in Prague. Grace, whenever you're ready, I'll hand it over to you.
Thanks, Jen. And thank you everyone for joining us in today's webinar. And let's go ahead and start. And I'm going to be spending just doing a little introduction as Dr. Vaklovic will be presenting the heart of the solutions that we are going to be presenting today. So the reason for the developments and leveraging technology, current technology that we have around is this is a common problem that we all see in dietary supplements, is the challenges that we have in product integrity, misbranding, mislabeling, and adulteration. So there's often, we've seen in the news some warning letters about misleading claims or false information in packaging, and also the inaccuracies in ingredient list, and the content claims are a little off. And of course, the adulteration concerns, where we actually have, we've seen pharmaceutical adulterants in the products. And these are a big concern because there's health risks related to misbranding. When you have undeclared pharmaceutical ingredients in supplements, they can cause severe health consequences, and also on the other hand, product recalls. And it's the same concern that you have when the dosage labeling is incorrect and the content claim is not what you thought it was, and there is a huge potential of harming our consumers. And along with that, of course, the regulatory enforcement actions that you potentially face. The warnings, fines, and mandatory product recalls, and there's usually is not very good for brands. And one of the preventive quality measures that always helps mitigate all these risks and keeps everything in order and transparent, routine testing and third-party verification gives some level of assurance that we are avoiding misbranding and mislabeling. And to building consumer trust, we usually want to always continue monitoring and testing and identifying our hazards. Besides it being a concern for product safety, it is required by law, and that we have looking into the contaminants that are possibly present in our products, and it will reduce your warning potential for getting warning letters. And so the one way of building the trust with the consumers is when you have your comprehensive testing, you cover all your safety compliance, and also you strengthen your brand reputation as we protect our consumers. Currently, the analytical trends, one of the things that we've looked into is what we call the non-targeted analysis, and then where we lead into suspect screening, flow, and targeted analysis. In the past, most common testing that we do is targeted analysis, where you know what it is that you're after. For example, if we've heard just recently in the news, some news about lead in your in protein powders. So things like that, those are usually targeted analysis, where you focus on a particular analyte and you go after that analyte. And where this trend is going is all the emerging contaminants, residues, and packaging migrants that you usually don't think of. And it's very challenging to figure out which one to target next. And so what the trend is that we've come up a solution, one of the solutions that we've come up with is what we call a suspect screening workflow, where you can have a group of contaminants, or like, for example, in pharmaceutical adulterants, you can have a group of possible contaminants, opiates, that are not supposed to be in the dietary supplement, but sometimes they make their way there, and you can screen for a whole class of them. So instead of screening and focusing on just one particular compound, you can focus on a whole class of them. So bringing the number of analytes that you're looking at to hundreds. And then if you want to go further, if you just want to do a total, you know, what could possibly be in there, then you go wider, where you don't even target. So anything that you can ionize or that can have a response, you can look at it. So the picture gets bigger and bigger. And the uses for this that we've is the, like I said in my example, the detection of unexpected adulterants. And you can also do fingerprinting of, for example, you have a new botanical product or new botanical ingredient that you have. You can do fingerprinting of that by running it. And Dr. Baklovic will give you a more detailed explanation of how this is done later. And also one other thing that could really be useful for is when you start with a particular ingredient, and you can look at that ingredient throughout the entire process and see what the chemical changes is for associated with processing. Helps with determining stability and shelf life. And so the advantages of doing the non-targeted suspect screening is that you can identify, you're proactively looking for possible adulterants and possible emerging contaminants. And because when you do a non-targeted screening or a suspect screening, you generate a lot of information about your product or your ingredient, and it helps in the decision-making process, but with data to back it up. And because you can target and focus and find for hopefully making the look for the contaminants, searching for the causes of the recall will help with alleviate management of recalls. And so with that, I am going to send you to Dr. Baklovic, to Lucas. to give you more of the details on the non-targeted analysis.
Okay, thank you, Grace. Hopefully everybody can hear me. Sorry for the delay. I was struggling to show my screen, but we should be all good now. So in the second part, we will look at some of the LCMS solutions that we have available. I will provide a brief introduction to the technique we will focus on mainly, which is liquid chromatography, mass spectrometry. And I will also characterize and highlight the differences in different LCMS-based workflows that enable targeted analysis, suspect screening, and also non-targeted analysis. I'll provide some examples of applications of these workflows in the area of dietary supplement testing. And in particular, I'll focus on screening and quantitation of pharmaceutical adulterants in these matrices. So mass spectrometry plays a central role in the testing of dietary supplements. It is a powerful analytical tool capable of analyzing highly complex samples with really exceptional specificity and sensitivity. It allows us to both identify and quantify trace compounds, which can be contaminants, but also abundant compounds such as active ingredients. When coupled with liquid chromatography, LCMS can provide efficient analysis of thermally labile, polar, and mainly non-volatile compounds, which are very common in botanical extracts and nutraceutical matrices. These techniques are ideal for detecting pharmaceutical adulterants, bioactives, emerging contaminants with high confidence. In addition, gas chromatography, mass spectrometry complements this by targeting or allowing analysis of volatile and semi-volatile analytes. While ICP-MS or inductively coupled plasma mass spectrometry is used for analysis of elemental impurities, heavy metals, and so on. Coupling of liquid chromatography with ICP-MS represents another powerful combination of techniques which basically combines chromatographic separation with elemental detection and allows speciation analysis, meaning that we can distinguish between different chemical forms of the same element, such as arsenic, chromium, or selenium species, which may differ dramatically in terms of toxicity or nutritional relevance. So these mass spectrometry platforms give us comprehensive analytical capabilities to ensure authenticity, safety, and compliance of dietary supplements. Since we are focusing on LCMS, let's take a quick look at how LCMS actually works. The process begins with liquid chromatography, where compounds in a complex sample are separated based on their chemical properties, such as polarity and interaction with the chromatographic column. Typically, different compounds elute from the column at the different retention times and are introduced into the mass spectrometer. Next comes ionization of these compounds, where molecules are converted into charged particles or ions, typically using ionization techniques as electrospray ionization or atmospheric pressure chemical ionization. And this step is crucial because only charged species can be detected by the mass spectrometer. Finally, in mass spectrometry detection, these ions are separated and analyzed according to their mass to charge ratio and generate mass spectrum. The mass spectrum provides a molecular fingerprint of the detected compounds and allows us to identify and quantify the compound, but also confirm the structure, detect presence at trace levels, and so on. So Together, the chromatogram and mass spectrum provides a powerful, I would say, multi-dimensional view of what's really in the sample. That said, not all mass spectrometers are created equal. Different instruments give us very different views of what's actually in the sample. So when we look at unit resolution mass spectrometers, such as single quadrupole, triple quadrupole, or ion trap, these are typically operated in a targeted way. So we do have a list of compounds we want to analyze for, and we have very specific settings when it comes to their detection. So we are looking only at those compounds. So in this picture, you can see we are looking at the house, but we see only certain sections, like, you know, the front door or windows. So targeted unit resolution MS will give us answer on presence, absence, or actual concentration for a discrete targeted analyte list. When we move to accurate mass, high resolution mass spectrometry, this allows us to open door to the scope of what we are looking at. We can collect data for way higher number of compounds, and we can also look at these signals with high accuracy, which allows us to determine potential elemental compositions of detected ions, compare the data to lists of compounds you want to screen for, even without availability of analytical standards. So as you can see, we are getting a broader picture of what's in the sample, not necessarily everything, but you know, we see more than with targeted methods. That said, you know, there's a trade-off. So if we are opening the scope, we are typically losing some of the sensitivity. So some of the low levels may not be visible using this approach. And eventually we can also switch to accurate mass, high resolution mass spectrometry with advanced software tools that allows us to perform non-targeted analysis workflows and comprehensively characterize the composition of the sample. And that's the real strength of high-resolution mass spectrometry. It doesn't just confirm what we know, it helps us to discover what we didn't expect. So we can actually find out that, you know, this nice house has some unpleasant surroundings, which kind of symbolizes potential problems in your sample, hidden contaminants, adulterants, and so on. So this slide just further highlights the differences between targeted suspect screening and non-target analysis workflows. These workflows represent increasing level of analytical scope and discovery. In targeted analysis, we already know exactly what we are looking for. We have reference standards, optimized analyte-specific acquisition parameters for our LC-MS analysis, and well-defined identification criteria. So the question for targeted analysis is simple. Is a particular compound present in the sample, and at what concentration? So we are looking at known knowns. These are our analytes with high sensitivity and accuracy and quantitative abilities. In suspect screening, we don't have reference standards for every compound, but we do have a suspect list. This can be a database of possible analytes that may be present in the sample. It can be a very long list. And we are collecting generic full scan data and search it against that list to look for matches, and this helps us to detect known unknowns, compounds we expect could be present, but we haven't confirmed yet. And finally, non-targeted analysis takes the broadest approach. We ask what compounds are present in the sample at all. There are no predefined targets. We have to rely on data mining, chemometric analysis, and structural interpretation to discover unknown unknowns. So we are basically trying to characterize the components of the sample. When we talk about these approaches, it is important to remember that what we actually detect and identify is typically only a small fraction of what's truly present in the sample. So the actual compounds that will be detected depend on many factors. It will depend on how or which extraction procedure you use, how much cleanup you apply to your extracts, what kind of separation system you use that will influence the type of compounds that will be retained and eventually eluted from your LC system, what technique and what conditions you use for ionization that will determine whether the particular compound will provide or will be converted into an ion and provide a signal. Overall sensitivity, mass accuracy, and additional information, as well as the workflow for your data processing and tools you have available for data interpretation. All these will affect how many of the compounds will be able to be detected or identified. So this is just a high-level overview of suspect screening and non-targeted workflow. As I said previously, we are trying to characterize the composition of the sample. So our sample preparation workflow involves some really generic extraction to discriminate or not to avoid any discrimination of sample of compounds that are present in the sample with no or very minimum cleanup. In these workflows, we typically combine different separation systems that allows us to analyze compounds of wide range of polarities, from extremely polar compounds to non-polar compounds. We may use combination of ionization techniques. Use of high-resolution mass spectrometry is a must. A very important step is data processing and pretreatment, basically data mining of the resulting data to extract as much information on detected compounds as possible, to merge signals that are coming from single compounds, align the actual signals we see across the samples, assign them with retention time, and we may also include some additional operations to center or scale the actual data set. And in the data analysis, we can restrict what we are looking for, what we typically do in the suspect screening workflows. So we may eliminate signals we want to further evaluate, or we may use statistical tools to actually highlight signals that are of particular interest to us, depending on the study we are performing. So we can compare different groups of compounds, different groups of samples to highlight different signals and look further into those. So when we have our signals of interest, the final step is actual identification. So we are trying to annotate and assign the actual signal in our LC-MS chromatogram identity. So then we really leverage the high-resolution mass spectrometry data, and we have to use wide range of resources, including spectral libraries, databases, and so on. And once we have our tentative identification completed and we want to confirm we performed identification correctly, we always need to use reference standard and collect data on the reference standard and compare that to the actual experimental data in the sample. So, as I said, choosing the right mass spectrometric tool is really important. There's a range of instruments on the market that allows you to perform suspect screening and non-targeted workflows, but we are actually using the latest instrument from Thermo Scientific, which excels at sensitivity, provides high mass resolution that allows us to measure ion signals with really high mass accuracy. So the actual data collection workflow is to operate in full MS when we are actually trying to get molecular ions of different sample components, but we also want to collect so-called product ion spectra, which are crucial to provide or to gain insight into the structure of actual compounds. So we are combining basically full MS data with data-dependent product ion scan experiments, and we are trying to collect product ion spectra for as many precursor ions detected in the full MS mode as possible. And then we use this data to assign elemental composition. When you measure at high resolution, there's only a certain number of combinations of elements that can match your signal in the full MS mass spectra with given error. So we are leveraging that high mass accuracy. We can get probable elemental formula. We are also leveraging the ability to evaluate the isotopic pattern of the particle ion, and then we can look further into the structure and identification based on product ion spectra. And we are using spectral libraries that are available, and we compare it to the experimental data. And in case we are failing using this approach, we can also assign some tentative candidates based on fragmentation rules and so on. So both MS1 or full MS data and MS or product ion scan data are vital for identifications. And depending on how we are successful and how much data we have available for a particular signal, we can perform or achieve certain level of confidence in our identification. So this classification of identification confidence level is according to Szymanski et al. You can see it goes down from having information on the exact measured mass of a signal up to confirmed structure. So with suspect screening and non-targeted analysis, we are actually we are typically able to achieve level 3 or level 2 identification, depending on the data we have available and what's available in our libraries. So this kind of underlines the need to obtain that product ion spectra for as many compounds as possible, and there are actually algorithms available that allow you to collect data for these components really comprehensively. Typically, instruments are limited by their acquisition speed. So if you have multiple signals being ionized and detected at the same time, you can collect the product ion spectra for a limited number of compounds. But using advanced algorithms and workflows, it's typically possible to get product ion spectra for most of the signals that are detected above a certain threshold. So again, this is crucial to be able to characterize the sample components as much as possible. And as I mentioned, it is really important to have advanced data processing and data interpretation tools. In our lab, we are using Compound Discoverer software, which allows you to customize your data processing, data mining, and alignment workflows to actually fit the purpose of your actual analysis or study. Depending on what you're trying to achieve. It's nice because it has integrated set of tools that provide some additional resources when it comes to identification. It allows you to easily review the outcomes of your identification attempts and so on. It also has a suite of statistical tools that allows you to highlight signals of interest based on statistical analyses, and you have additional options when it comes to databases, different lists, for example, in your suspect screening workflows, use of internal standards, and so on. So talking about spectral libraries, I want to highlight two of those that are great resources. One of which would be MZ Cloud Library, which is a highly curated spectral library with MS1 and multi-stage fragmentation spectra for over 3,000 compounds. It's being further updated. So that's a great resource for tentative identifications of your signals you see in your samples. We also do have NIST LCMS library, which brings data or fragmentation data for approximately 50,000 additional compounds from diverse groups of analytes, including contaminants, metabolites of plant or human or mammal origin, and so on. So, these resources allows you to assign a probable structure. In other words, identification confidence level to your compounds of interest. And if you fail to annotate your signals using spectral libraries, there are additional tools that give you some additional hints when it comes to potential structure of your compound of interest. And these would be called MZ Logic and fish scoring. Fish scoring allows you to perform so-called in silico fragmentation on your candidate structures and basically look how well those fragments that are observed in your product ion spectrum match your experimental data. So again, great research that allows you to assign at least tentative structure in your identification process. And this is just an overview of what you can see and how nicely integrated you have all that information when you're looking at a particular compound. In this case, it was sibutramine, which is a weight loss drug, which was detected in dietary supplements. And you are getting a number of information ranging from elemental composition, how accurate that particle elemental composition aligns with your exact or measured mass, how well the actual isotope pattern fits with that particle elemental composition, and what are your hits with different spectral libraries. So that's extremely useful, and trying to chew through huge amounts of data you will typically get through or from those workflows. So let's switch gears now a bit, and I also wanted to highlight some of the target analyte capabilities we developed. And here's just an overview of usual suspects when it comes to safety and authenticity of dietary supplements. Now, not all of these compounds will be able to be addressed by LCMS. Most of them could be, but my focus in this webinar would be on adulterants, particularly pharmaceutical adulterants. So dietary supplements are typically perceived by the consumers as safer, more natural alternatives to conventional medicines. Unfortunately, this trust is sometimes exploited through fraudulent, profit-driven adulteration practices, as mentioned by Grace already. In these cases, manufacturers intentionally add active pharmaceutical ingredients to boost or mimic the biological effects that the supplement is marketed for, whether that's weight loss, sexual enhancement supplement, pain relief supplement, or other type of supplement. These adulterants can include approved prescription drugs, substances withdrawn for safety reasons, or even unapproved designer analogs or different modified chemical variants in order to avoid detection, basically. What's especially concerning in these is that these compounds are often present at concentration near or even above therapeutic doses, so really high concentrations. That means that consumers may unknowingly ingesting pharmaceutical level amounts of potent drugs. Such adulteration carries serious health risks, obviously, particularly for vulnerable populations or individuals that take other medications. So in the United States, the FDA plays a crucial role in monitoring dietary supplements for adulteration with pharmaceutical ingredients, and this problem and fraudulent practices are clearly recognized by the FDA. Through its health fraud product database, the FDA tracks products found to contain active pharmaceutical ingredients that are not declared on the label and are being illegally added to enhance or mimic drug-like effects. So since 2007, the database has accumulated over 1,200 entries, which kind of documents supplements adulterated with these substances from a wide range of different classes. You can see which adulterant or pharmaceutical ingredient classes are most frequently used. What's important is that the actual cases represent only the identified fraction of occurrences, meaning that the true extent of adulteration in the global supplement market may be actually much higher. So, as I mentioned, really high concentrations. Another concerning fact is that in many cases, multiple pharmaceutical ingredients are actually present in the adulterated products. So looking at the targeted methods we developed at Eurofins, we worked from a target pharmaceutical list defined in the NSF-NC most recent standard document that covers these 4 classes of adulterants. Our methods are based on targeted LC-MS technology that basically targets at fast screening of the compounds. You can see that our target list range from 10 to several dozens of compounds in a single run, using high throughput approach, while allowing us to confidently detect and identify these target compounds. Our methods were fully validated in terms of screening, using evaluation of probability of detection and identification, but also were shown to provide acceptable quantitative data. We do have a strict detection and identification criteria, and we also have a number of QC measures in our routine testing. So typically we screen our samples against a single level solvent standard injection at the reporting limit. If we see any hits, we will reanalyze the samples if needed, and we quantify using either solvent-based or matrix-based standards. And as I already mentioned, we do have quality control injections. We run many of the samples actually fortified with the target analytes to demonstrate these are recovered appropriately, and also we heavily use isotope labeled internal standards for both quantification and monitoring of the extent of matrix effects that occur during the analysis. So this is just a high-level example of what we actually see in our testing. This particular example focuses on joint health and pain management supplements and sexual enhancement supplements. As you can see, we detected acetaminophen, for example, which is a common over-the-counter pain reliever that was present in botanical gummies at extremely high concentration. All of these actually, when recalculated to the serving size, are well above therapeutic doses or daily recommended therapeutic doses for these respective dietary supplements. So that diclofenac is a non-steroidal anti-inflammatory drug. As you can see, in this case, glucosamine and chondroitin powder that are frequently used to promote joint health, has been adulterated with this active ingredient. We see also other compounds such as dexamethasone, prednisolone, piroxicam, or hydrocortisone. Similar situation with sexual enhancement supplements. In this case, we detected PD5 inhibitors, which are also known as erectile dysfunction drugs. Again, at really high levels. And among the actual parent compounds, we often also see analogs, which are basically modified compounds. So, for example, aminotadalafil has an extra amino group attached to the tadalafil moiety. Again, these compounds have no safety profile. These are analogs that were never clinically tested. So, there's relatively high risk associated with exposure to these compounds at high levels. So let's look at suspect screening workflow. At this point, we evaluated this workflow using high-resolution mass spec. So we basically took our full list of pharmaceutical adulterants that we test for in our targeted methods, and we selected some particularly complex finished supplements and also ingredients such as botanical extracts, and we fortified those materials with all these compounds, and we collected data using some appropriate conditions to retain and detect most of these compounds, and we employed the compound discoverer software using a customized workflow to screen for these compounds without any actual prior knowledge or simulating basically non-targeted analysis workflow. So, the data processing workflow was designed in such a way that there were no actual target list. And we looked at the data from different perspectives. We analyzed in both positive and negative mode. These are just example chromatographic records obtained for these samples, and you can see these are quite complex. There's a number of signals already seen in the total ion current chromatogram, and each of these peaks may represent a compound. Many of the additional compounds that are in the sample are actually hidden below and represent way smaller signals. So, across these two samples, ionization mode, we were able to extract or data mine over 1,500 compounds. And then we actually looked how well we did in that workflow. So particularly, we also tried to evaluate which of the actual compounds spiked were detected or not. And of the 120 compounds that we actually fortified the samples with, we could see around 110 compounds. Some of the compounds just gave too low signals under the conditions used, or gave some or had some coalitions with isobaric compounds, so could not be identified in a unique way. But we were pretty happy what we saw in these experiments. And all the detected compounds could be actually identified with relatively high confidence level, with probable structure assigned or probable compound assigned based on comparison to spectral databases or some additional tools we had available in our software processing, in our processing software. And the way we collected data allowed us to look at also additional compounds, not only pharmaceuticals, but we could detect a number of additional sample components and also assign them some tentative identities. In this example, you can just see identification of curcumin when you in the upper and lower side of the mass spectrum, you see a comparison of experimental data and the data or spectral information available in the spectral library that was used to match and assign tentative identification. So just one example, but you probably understand the potential of this workflow to characterize composition and look deeper into composition of the sample. So we are currently working on our routine offering. This is pretty complex to provide or offer in a routine way. We are currently working on suspect screening, offering to screen for several thousands of potential adulterants, and we are working with compounds that are actually included in one of the spectral libraries we have access to. Steps we are working through are defining our chemical space to kind of demonstrate that our method is able to capture and detect wide range of compounds that differ widely in terms of their molecular weights, polarities under which they provide signals, and so on. Huge effort is associated with optimization of the LC separation, so we can retain and separate as many compounds as possible. And as I mentioned already previously, you need to typically employ two different conditions designed to provide retention for more non-polar compounds and also for highly polar compounds. So once we work through all these optimizations, obviously we need to perform validation for representative analytes to evaluate false positives, false negatives. These representative analytes need to be a good representative set of compounds to cover all different properties of our intended analyte scope. And there are also QC, QA considerations, reporting considerations to be addressed to bring this offering online. This particular offering is intended for pharmaceutical adulterants, but the same workflow could be also implemented to offer screening of different classes of compounds. Obviously, it would require additional effort, additional development and validation, but the actual workflow would be applicable to a wide range of compounds that actually give signal using the LCMS technique. And now let's take a look at some of the non-targeted analysis applications. Some of these were highlighted or discussed already, but besides screening for new contaminants, residues, potentially also for packaging migrants, you can also use or look at your samples in a more comprehensive way. Evaluate the actual sample fingerprints. Some of these potential applications were already highlighted. By GRACE, you can fingerprint your ingredients or materials and look for different markers of quality or various active compounds. you can try to identify those, look at additional components of your samples, and also monitor chemical changes that may be associated with processing and other procedures. So let me show you a few examples. This is pretty simple example that demonstrates identification of some impurities in a raw ingredient used in dietary supplement production. In this case, it's beta-alanine. You can collect the data for a blank that is basically a solvent used for extraction of your sample, and you can run actually the sample of interest, and you can look into different chromatographic peaks you see in the record. So we see one huge peak, which would be the actual active compound or the main constituent of beta-alanine powder, which is beta-alanine. But we also see some additional 2 peaks of interest. Are these impurities? Are these coming from the background? They're not coming from the background, as we don't see those in blank record, and this blank was processed the same way as the sample. So these are likely associated with the active ingredient, and actually we are able to look at those peaks and extract spectral information, and using the tools and procedures I showed earlier, we are able to assign tentative identities for these, information, tentative identification of these compounds. So you can see, it's actually a dimer of beta-alanine and some other compound, and we actually confirm these based on analysis of reference standards to support actual identities, and we're able to report this identity of the impurities in that particular sample to the client. So another example, you can kind of monitor consistency in terms of chemical composition of your ingredient batches. So, we are collecting fingerprints in the same way for 3 different batches, and we are getting our LCMS chromatograms, and then we collect data for additional ingredients, and we can use statistical tools to highlight differences between our actual ingredients. So, for example, these 3 batches were processed and analyzed in the past, and then we got a new ingredient batch, and we are always comparing this new batch to old batches to confirm consistency. And this comparison can highlight some differences and signals of interest that you can look further into using high resolution LCMS. So in this particular example, an unknown peak was highlighted using differential analysis and identified as an actual pharmaceutical that was present in the sample, chlorpheniramine, which is an antihistamine drug with some sleep aid effects. So this particular example shows you how fingerprinting can help you to identify or highlight unexpected compounds in your materials. And in this particular example, we looked at single compound, but we can also look at the full fingerprints of the samples, and using multivariate data analysis tools, you can also look into the structure of the data. So in this particular example, we are using principal component analysis to evaluate structure of the data. In this particular example, there were 3 different ingredients analyzed, and you can see that based on the type of the ingredient, these samples actually cluster together, indicating that they have similar fingerprints. And in addition to clustering of the sample, you can also look into actual peaks or compounds that are responsible for these differences in so-called PCA loadings plot, and you can evaluate each of the detected signals and actually look at abundance or response profile across the group of the samples, which can help you understand what are characteristic markers, what are changes, differences in your data set. So this kind of brings additional potential applications in the area of dietary supplements and many others. Additionally, if you switch to using so-called supervised pattern recognition techniques, and you have a sufficient number of samples, you can also develop some classification models that allow you to classify an actual unknown sample into different classes. So there are certain caveats to this approach. Your sample set you use for training of the classification model needs to be really robust, and so on. It brings additional challenges when it comes to collection of the data and compensation of batch to batch or analytical batch to analytical batch variances, but can be really useful and powerful tool to ensure quality and authenticity of your materials of interest. So in conclusion, hopefully we showed that LCMS-based suspect screening and non-targeted analysis workflows can be really powerful and open door to many diverse applications that are relevant to the field of dietary supplement testing. So you can look at contaminants, residues, adulterants, or migrants, but you can also explore comprehensively composition of your sample, look into differences and changes of your material, and get valuable data for interpretation of different challenges. And I also want to state that although we have all these advanced tools available, targeted LCMS methods still remain really, really important tool and gold standard when it comes to quantification and routine analysis of different analytes at low levels. I would like to acknowledge my colleagues at Eurofins, and I also want to acknowledge our collaborators at Thermo Scientific, which helped to generate some of the data I showed today. I will thank you for your attention, and Grace and I are looking to any questions you may have. Thank you.
Lukasz, thank you so much for that presentation. We're right at time, so unfortunately we won't be able to address any questions, but we definitely will follow up with those who ask questions later on following the presentation. So I just want to thank everyone, including Grace and Lukasz, for the presentation today, and a big thank you to everyone who's attended. Definitely reach out with any questions, and we're happy to connect further to discuss this very important topic. This now concludes today's presentation. Have a wonderful day.